Okay, hello everyone. My name is Shipan. I'm come from University College London. Today, I'm very excited to share our work. We're using the single-cell language model to estimating the cell state during the epithelial to mesenchymal transition. And the target problem we are talking about here is epithelial mesenchymal transition, which is a quite important problem in cancer research. These transitions, so let's just short it as EMT, is a kind of widely considered biology problems in cancer research. The key difference is between this transition, it gives the cancer cells the capabilities to go inverse into different tissue types. And the difficulty part is you need to identify during this transition, the intermediate state, how it looks like. In the beginning, researchers imagined that, well, intuitively, as a binary transition problem, which is shown in these figures figures 1. And so where's my dot? Yeah, as this one. You can see that, you can imagine that the cancer cells from the beginning point and during the transition to the mesenchymal stadius, and then go to the reverse back to the very beginning point. But actually, when you dive into the data, you will see there's multiple cases like this one. Your transitions go to totally different routes, and the intermediate cell state would be various. And in some extreme case like this one, you can see that the beginning point, the epithelial point, and the ending point, which you assume is the mesenchymal point, are totally different. So, which makes this problem quite difficult to identify this intermediate state. Now we have a lot of large language models in this conference, and today we are trying to say that the tool we are using is slightly different, which is a single-cell large language model. It's not trying to get the string as the input, but consider the kind of more like a tabular data with some particular design. It's also using transformer architectures and gives the using the masked self-supervised training paradigm, but it's not totally different with the genomic language model or protein language models. By following this work, researchers are trying to say that this single-cell language model can eliminate batch difference. It can capture very complicated cell type. And it's actually learned the potential distribution of the single-cell data format. So we are asking question, can we try to use this method to identify the intermediate cell states in our research problem? So we're trying to organize a kind of data from the public available data sets, which involved 4 different tissue types and all simulated by TGF-beta. The cells are sequenced for different time points from day 0, which is not treated by TGF-beta signals. And after 8 hours, day 1, day 3, and day 7, they are all sequenced with single-cell resolution. So we can define these as before the treatment is the cell will more close to the epithelial stadius, and after 7 days is more closer to the mesenchymal stadius. So we try out other people's methods. The left-hand side we show here is the visualization for the embedding space for the raw data. As you can see that there are very clear 4 clusters, which shows there are 4 different tissue types. And the SCBERT actually do quite well to eliminate the batch difference, but it's not show anything about our problem, the cell intermediate states. I would say that it's actually doing the right work because this model are trained for the cell type classification, and all of these cells supposed to be the cancer cells. For our model, we involved 2 training phases. And the first one, we are trying to train the experts net, which for each independent cellular states. And then we trying to fuse all of these experts nets together with the commonly used classifier or regressor layers. And we achieved a fairly good performance if you compare with traditional machine learning algorithms such as KNN, Random Forest, AdaBoost. And for each independent cellular state or the independent cellular point, it's more like we got all achieved a good performance. So let's back to the embedding space and see how it goes. So you can see that our model successfully clustered different time point cells into different clusters. We color them from day 0 to day 7 here. And because we train our model with discrete labels, which means that we don't tell our model these categories are temporally correlated to each other. But in the embedding space, it's naturally one after the other from 8 hours, day 1, day 3, and day 7, which means that our model actually capture some biology knowledge behind it, behind these distributions. So we do further validation on the independent data sets. To be fair, this is quite difficult because they choose different cell lines, which is different with our training data, and they sampling their cells with different time points as we introduce. We only use the time point before 6 days because after 6 days, they will just remove the TGF-beta signals. So here we also start with embedding. So this is a fate embedding for the row data points. As you can see that there are several different colors. I listed them from T0 to T7, which makes it more easier to understand. We predict a continuous score for it. As you can see, that's from the darker to brighter. We do capture these changes during the time point. And well, if you see this violin plot, you can see that for different categories, this distribution of the predicted score keep rising during the time point, which is quite good because the wet lab design are quite different, to be fair. And we actually do more validation for the external EMT inhibition data sets, which is from the single cell, which is another data modality. In the left-hand side, we show the box plot about the overall predicted scores between categories. Here are 2 patients, and each of them have 2 slides. One is before the treatment. Another one is they are treated with EMT inhibitor, which is called Nation 1. So you can see that the predicted score are getting lower after the treatment, both of them. And if you see that visually, you can see that more brighter dots in the before treatment part and more darker in the after treatment part. So which means that our model can capture these EMT signals in the different modalities. So the key takeaway is we actually create an EMT language model which allow us to capture the discrete states or estimates a continuous score for the EMT continuums in the single cell data as well as in the spatial transcriptomic data. Also, we achieved the quite excellent performance, which is reported as AURC, it's 90%. And well, we have more work we can demonstrate if you are interested with or you can find in our preprint papers. We developed the DESI score to extract the important genes from our model. And we have actually more validation data sets if you're interested with. So thank you for the team. And I also put our GitHub link here. So if you're interested, you can just go to the GitHub. We will upload our preprint later. And if you're interested, you can also email me. Thank you.
Thank you very much. So I will ask in the meeting room if there are any questions. Online, we don't have any questions. Any questions? Thank you very much.
Yeah, sure.
And, sorry, there is a question from Jan. No pressure, please.
It's nice to get a question.
Thank you.
So I was just curious to know if you'd looked at different developmental EMTs across your data. If you'd thought, for example, about gastrulation or neural crest migration and how that then links into the states you're seeing in this EMT continuum.
Sure. If I can go back a bit. So basically for our validation and our experimental data, For the EMT we define is totally based on the wet lab design from our data sets. And they designed that to do the treatment in some time point and sampling their sequencing their cells in a different time point. So we decide that for the after actual time goes, the cell will naturally transform from epithelial to mesenchymal. So there must be some dominant intermediate states in these different time points. So it's not actually we have any design on it, but only for this wet lab based. So it's actually from their own paper, openly available.
Okay, I was asking about developmental EMTs because of course you're working with cancer.
Yeah.
So for example, gastrulation is a canonical EMT that occurs in development, and neurocrest migration is, and there are different markers involved in those different states. So I was curious to see if you'd thought about overlapping your interesting data and your interesting model with these other types of EMT, you know?
That can be a very interesting future work for that. But for the currently we have is totally based on the single cell. So that's just like that. Yeah.
Okay, thank you.
Thank you very much.